Hello, everyone. It's great to be here, and I'm really glad to be able to introduce the second edition of the Gut Microbiome Health Index Challenge. I'll start by giving a short introduction to the topic. So if you haven't joined us this year, it will be a good introduction to you to get to know what it is all about. Then I will briefly tell you about the submissions that we had last year and the interesting insights that we had. Then I will continue to telling you about the updates to the challenge this year because we changed it slightly. And then I will give you an overview of what to expect in the next talks so that you get excited about the submissions. Right, and this is not changing. And I don't know why. Thank you. Okay, so why would we want to evaluate our health from stool? You might have heard of the gut-brain axis, which is a phrase that stands for the connection between your gut and your brain. And it basically means that your gut has a huge influence on the rest of your body, not in terms of your health, but also in terms of the way you behave, the way you think. It also influences your moods. So it remains a chicken and egg question whether it is the microbiome that changes in disease or whether it is the disease that is changing the microbiome. But we do know that the microbiome does change in the advent of disease. So we can use this information for the diagnostics. Actually, microbiome-based diagnosis is becoming a really good alternative to other methods such as blood collections or biopsies because it is becoming more affordable, it is non-invasive, and it can also be collected regularly. There are a number of methods that are currently allowing you to evaluate your health based on your stool samples, and there are two classes. So the first one is purely based on diversity, alpha and beta diversity, which are measures of your gut microbiome diversity and richness. The general idea behind this is that the more rich, the more diverse your microbiome is, the healthier you're supposed to be. So of course, the higher your diversity, the healthier you are. The second class of indices is slightly more complicated. Those indices are based on taxonomy. So what they do specifically is they look at the contributions of different bacteria, so health-associated beneficial bacteria and disease-associated pathogens, and they calculate the relative ratios. So if you have more beneficial bacteria than pathogens, then you're supposed to be healthier. And of course, you want to minimize the number of pathogens that you have. So this is all based on very simple ideas. But why do we think that we need a new index? Let's have a look at the recently updated definition of the microbiome. What it tells us is that the microbiota, so the community of microorganisms such as bacteria, archaea, viruses, are just a small part of the microbiome definition. And it is the whole theater of activity, so all the functions that those microbes have, all of the metabolites that they produce, their structural elements, even the environmental niche that they are present in, that fully define what a microbiome looks like and what it behaves. So if we are restricting ourselves just to the microbiota, as we've shown here, just the way that the other indices are doing this, we are really missing out on the important aspects of this microbiome that we should be looking at. There's also another reason that we should move towards functions instead of relying purely on taxonomy, and I'm presenting it here. The top left figure in blue is showing the distribution of species in a healthy population from the Human Gut Microbiome Project. And we can see that the majority of species are present in less than 20% of individuals. So if we are trying to define health-associated species, it is really difficult because we all have different species, and we would not be able to identify those taxa that are present in all of us universally across the world. Whereas if we look at the distribution of functions, which is shown in the figure B, we can see that indeed there are a lot of functions which are unique to specific populations, but also there is quite a high prevalence of many functions which are present in all healthy microbiomes. So this was only done on the Human Microbiome Project 2, which was one cohort, but then we extended this analysis to other healthy people, and you can see at the bottom that the trend remains unchanged. So we can see an enrichment of functions present in 80% of individuals, at least 80% of healthy individuals from various cohorts, and we can use this information to start building our index. And so our index, the Q2PD, which is a shortcut for Q2 predict dysbiosis, initially implemented as a CHIME2 index, so this is the way we formulated it, right? So we started by determining features associated with health. Those features include information such as the number of those core features found, the interactions between bacteria, their contributions to different health-associated functions. We then passed all of those features to the random forest model, and we output a score, which is basically the score that our model predicts, right? So the 0 is absolute disease, and the score of 1 is perfect health. So let's now see how our index compares to the other indices. We can see our index. I do not have a pointer, unfortunately, but I'll just try to describe it. So the top left figure is showing the results from our index, and then the figures on the right, the smaller ones, are showing the results for the other indices. We have 3 different cohorts, hence we have 3 different groups of bars, and the green bars represent people who are healthy. So if an index is working correctly, the green bars are higher than the other bars, which stand for people with inflammatory bowel disease. We actually have 2 different subtypes, and people who are obese. So we can see that our index successfully distinguishes between those healthy and diseased individuals because the green bars are higher than the others. It is not the case for some of the indices. Actually, some of those are even showing the inverse trend, so they are wrongly identifying diseased individuals as healthy. We also performed some other metrics. So we looked at accuracy and general feasibility of using this index in a classification. Our index is shown in red at the bottom, and then the other indices are shown in the other colors. So we can see that, for example, in the case of accuracy and AUC, our index is outperforming the other methods on the IBD cohort. So we trained our index only on people with inflammatory bowel disease, so IBD individuals, but we wondered whether it would be applicable to other diseases too. And it turned out to be working really well, despite not seeing the other the other diseases. Again, our index is shown in red, and we're comparing the accuracy and AUC to the other methods. It needs to be noted here that actually the other methods had already been trained on some of those data sets, so they were familiar with the other diseases, whereas our index is not. So this is particularly exciting. And I'm also showing a matrix that presents different cohorts in rows and then different features that our index uses in columns. The color of the cells tells you how important each feature was in the classification of different diseases. So some of those features are taxonomy based, some of those features are function based, and we can see that depending on the cohort and disease, the importance of different features really changes. So it might give us some indication in terms of which information the index is using to classify people with which diseases. So in summary, we developed an index that was based on functions of the microbiome and bacterial interactions. We developed it purely on healthy people, and we validated it against people with IBD, so inflammatory bowel disease. But it also seemed to be working really well on other diseases, better than taxonomy-based indices, which had been familiar with those diseases. This was recently developed in Giga Science. So you're welcome to have a look at our publication. And now I'm going to tell you something about the past results from the CAMDA challenge. So the CAMDA challenge last year was supposed to build on the indices that I've just described and to use taxonomy and functional tables from the metagenomic sequencing, so microbiome data, to improve the performance of the existing indices. And what I'm showing you here is one of the most interesting findings. So the competitors developed the RFT GMHA. GMHA is one of the previous indices that was already available at this time. They added their own features, so they performed additional analysis to identify bacteria and functions that were relevant for the health versus disease classification, and they managed to improve the accuracy of classification to almost 90%. Their method also worked very well on the test dataset, which was a Polish dataset of COVID-19 patients. And despite not training their index on COVID data, it seemed to work really well in identifying those people who had COVID. What was particularly interesting from the biological standpoint is that they noticed that in general, all of the healthy samples had taxonomic changes, but the way the functions were changing were very much dependent on the disease, which was an indication that you could probably start changing those functions, that modify the functions that you're looking at in order to classify different diseases. And so we are at this year's CAMDA. We are starting from a slightly extended data set. So this time we provided over 1,000 taxonomic and functional metagenomic profiles. And we asked the participants to classify healthy versus disease individuals. We actually included a lot of different cohorts with a lot of different diseases, because last time we restricted this set to make it a little bit easier. So this data set this year is very, very diverse. And what we particularly wanted to highlight this year is the way the participants integrate taxonomic and functional information. So we didn't want to focus purely on improving the classification, but we are looking particularly for those novel and creative solutions of solving this data. And actually, we are really impressed with this year's solutions. The participants used a wide range of ensemble methods, so they combined different machine learning models. They also proposed some topology-based solutions, which are not commonly used in the microbiome field. And they also targeted their indices towards different applications. There was a very interesting case of developing a method on rare diseases. Interestingly, despite playing with different approaches from usually different fields, they still managed to improve the classification, which I think was really, really exciting, because probably without applying those new approaches, we wouldn't be able to see performance being improved so well. And so I'm happy to introduce this challenge, and I'm really looking forward to hearing the presentations of the participants.
Thank you. Are there any questions to Kinga about the challenge? Yep, there's a microphone.
Has anyone ever tested these indices on, let's say, patients that were, or people that were normal, classified as normal, but then subsequently developed a disease, like to see if they had some prodromal microbiome?
Yeah, of course. It's very difficult to create such studies, right? Because you're usually limited in terms of the samples that you can sequence, and then you'd have to commit to tracking those patients over time. So we do have data sets of people who are, for example, improving or getting worse. IBD is a very good example where you have flares of IBD from time to time. So we use this type of data as a proxy, but there aren't many data sets currently available which have this information. But I think it's really, really exciting, this early diagnostics. And I think this is also partially the way the field is currently going.
I could only add to this that, as Kinga said, that in this many features, sometimes we see the taxonomy is working better, sometimes the function is working better. So this will be about, understanding disease, whether it's originate in the gut microbiome, or maybe the gut microbiome is just a mirror of what is happening in the organism, like all the inflammations. So here, I think in case of the using it as a early markers, definitely the microbiome will develop this direction. There are still many things to be, let's say, validated and standardized, but this is in fact happening. There are more and more evidences where there is a for specific disease, there are clinical tests which are showing that clinical trials which are showing that indeed the specific taxons, they are related, but they are still, we need to move further to functions, because it's not always will be just the taxon is related, especially that the big problem is the between cohort agreement. We are living in different environmental biomes, which are influencing our health, and they are also influencing which species or which strains can be seen in our gut. Yeah, so then comparing just on the taxon between different cohorts from different parts of the world for the gut microbiome is very challenging. And as Kinga has shown, moving to the functions will simplify this. Yep. Yes, please.
Thank you for the introduction. You mentioned that in the second edition, you want to maximize the creativity of the solutions. And I think, I mean, I'm just saying, based on the very limited time that you presented the results, but it seems like there is not a lot of metrics to evaluate to performance, at least identifying whether a patient has a disease or not. So this seems like a binary classification problem. So the performance could be easily visible within some of the metrics. So how do you kind of evaluate this novelty aspect, is what I am saying, in comparison to these type of binary classification metrics?
Yeah, of course. I think there are many different answers that you could pose here, but maybe I'll start by saying that when we asked most of the participants to improve the predictions last year, most of the solutions were as follows. They would take the functional and taxonomy matrices, they would perform basic feature selection, they would pass them to random forest and notice that the results improved somehow. And it seems to be a very consecutive set of approaches, because this is what people are doing in the field. We're analyzing the microbiome, we are not applying any neural networks, we are not doing anything too complex. It usually starts by just applying a logistic regression or random forest and seeing where we get, right? So we wanted to see whether we could take something from the other fields and to look at it from a different way. So this was the main goal of the challenge. We said that the improved prediction is something that we are interested in, of course, but even as the participants has shown, there are different ways of looking at the data, and it doesn't always have to be focused only around just classification. The participants have found a lot of interesting insights in the meantime, and I think those are much more valuable. We just need to wait for the presentations because I think the findings are really exciting. But yeah, I agree. Health is not binary. The question is where we set the threshold and how we want to look at this problem.
Okay, one last question before we switch to the talks.
So I'm not familiar with the input you provided for participants. And having said that, I think it makes a lot of sense to move from taxonomy to function. But as far as I know, a lot of data sets that are out there are based on 16S. And how can you use 16S to infer function? You can, but it's kind of not very confident. Do you have anything to mention about that?
Yeah, so this challenge is not using 16S data. I think, well, I can't definitely confidently say that the field is now moving away from 16S sequencing, because I know that there are many labs who are using it, but I think there are different reasons for using 16S or metagenomic sequencing. And if we want to take advantage of functions, we just need to do metagenomic sequencing, which is also sufficiently deep. So I think this is at the moment the limitation. Yeah.
Okay, so thank you, Kinga, one more time.